Imagine walking into a massive library where every book contains instructions on how to build and maintain a living thing. This library is like your DNA, storing all the information needed to make you, down to the tiniest cell. But just like in a real library, you don't need every book at once. Instead, your body carefully selects, copies, and follows specific instructions to build proteins, the molecules that keep you alive. This process, called gene expression, happens in two main steps, transcription and translation. Let's take a closer look at how it all works.
What is gene expression? Gene expression is how cells take the information stored in DNA and use it to make proteins. It happens in two major steps, transcription, where DNA is copied into the messenger RNA, and translation, where that message is used to build proteins.
The first step, transcription, takes place in the nucleus, the reading room of our library. But instead of removing the original book, DNA, the cell makes a copy of just one chapter, a gene, onto a piece of paper, which is messenger RNA. It all starts when an enzyme called RNA polymerase finds the right section of the DNA book called the promoter. This signals where transcription should begin, and the DNA unwinds so the librarian RNA polymerase can start copying. As the librarian works, they write down a complementary RNA copy, replacing the DNA letters, which is A, T, G, and C, with RNA letters A, U, G, and C. This mRNA copy will serve as instructions for making a protein. Once the entire section is copied, the librarian stops writing and the new mRNA transcript is ready. Before leaving the nucleus, the mRNA gets edited. Unnecessary nodes, introns, are removed, and important sections, exons, are kept. A protective cover, a 5 cap, and a security stamp, poly A tail, are added to ensure the message is delivered safely to the ribosome, the cell's protein-making machinery.
Step 2 is translation, turning the instructions into a product. Now that we have our copied instructions, it's time to put them to use. Translation happens in the cytoplasm, where ribosomes act like skilled workers in a bookbinding shop, turning the written instructions into a final product, proteins. Initiation. The ribosome attaches to the mRNA and reads the first 3-letter word, called a codon. This is like the bookbinder picking up the first instruction. A special worker, transfer RNA, brings in the first building block, an amino acid called methionine. Elongation. The ribosome moves along the mRNA, reading each codon like a bookbinding manual. Each new tRNA brings the correct amino acid, which gets linked together like pages in a book, forming a growing protein chain. Finally, termination is when the ribosome reaches a special stop codon, and translation ends. The completed protein is released and ready to be folded into its final shape so it can perform its job in the cell.
Why is this important? Proteins are the final products of the entire process, and they're essential for life. They make up our muscles, enzymes that speed up reactions, hormones that send signals, and even antibodies that fight infections. Every function in your body depends on properties made by proteins. But what happens if there's a mistake in the DNA book? Mutations are like typos. Sometimes they're harmless, but other times they can change in the meaning of the instructions, leading to defects, proteins, and even diseases. Recap and closing thoughts. So let's review. DNA is like a vast library of instructions. Transcription makes a temporary copy, mRNA. Translation reads that copy and assembles the final product, a protein. This incredible process happens in your cells every second, allowing life to function. Just like a well-maintained library ensuring knowledge is passed down, our cells carefully control gene expression to keep everything running smoothly. This delicate system is what allows life to grow, adapt, and survive. It's amazing to think that every moment inside every cell of your body, an entire library of knowledge is being read and translated into the molecules that make life possible.